Welcome to today's webinar. My name is Jose Francisco, Project Manager at the IAS-USA. Today's webinar will be going over Bacteriophage Therapy. The enemy of my enemy is my friend. I'd like to welcome our presenter, Dr. Robert T. Schooley from the University of California, San Diego. Welcome, Dr. Schooley.
Thanks very much, Jose, and good morning or afternoon, depending on where you are today. I'm going to spend the next hour or so talking about an area that has been moving relatively rapidly after about a century of little progress, meaning the use of viruses to treat bacterial infections, bacteriophage therapy. This is a new area for IAS-USA and kind of the inverse of what we normally do. Here we're giving viruses to people rather than trying to eliminate them, hence the name of the seminar. This is a CME accredited seminar that has credits under AMA period category 1, as well as accreditation for the following for the activities shown on this slide. We're happy to have support from several partners in the pharmaceutical industry listed here. And we're now going to move to how to navigate the webinar. You'll see a series of poll questions off and on. You'll have an opportunity to, in this separate window, choose your response for the poll. Jose will then show us the results of the poll. We'll discuss them at the appropriate time and move on as we move ahead. We'll have a time for question and answers at the end, depending on the time. We're hoping to be able to get to all of them, but if we don't, we'll try to answer the ones that we can address and get back to you individually. To get you warmed up on the poll questions, just start on a fairly simple one here. How many of you have heard of bacteriophage therapy? You're very familiar, vaguely familiar, never heard of it, you have no idea what we're talking about. Please go ahead and vote, and we'll have opportunity to see how this plays out. So there we go. About a little over half of you have heard about it. A quarter of you know all about it. And very few of you actually have never heard of it. So hopefully we can whittle that last two categories down over the course of this morning. So let's go ahead and get started. At the end of this, we hope you can understand what advancements there have been in bacteriophage therapeutics over the last half decade. And we'll understand about some of the ways that bacteriophage therapeutics might play a role in clinical practice moving ahead for those of us in infectious disease, medicine, and surgery. So let's have a couple of pre-test questions. The first one is that relates to how bacteriophages kill their targets. Please have a look at the answers here and see which one you think is the most likely way that bacteriophages are able to kill their bacterial targets. They integrate the DNA into the host bacterium and downregulate RNA transcription. Do they actually instruct their hosts to synthesize lysins that disrupt the cell wall of the bacterium? Do they activate bacterial quorum sensing? Or do they resensitize bacteria to antibiotics to which they develop resistance? Please pick one, and then we'll see how the answers compile. Okay, so most of you felt that they are able to cause their bacterial host to synthesize lysins that end up disrupting the cell walls, with a smaller fraction thinking that they downregulate the RNA transcription of the bacterial host. Let's go on to the next question. We'll be getting to the answers of these obviously at the end of the talk. All therapies have some side effects. Which of the side effects might you expect with bacteriophage therapy? Induction of septic shock by accelerated lysis of bacterial pathogens, induction of bacteriophage-specific adaptive immune responses, massive disruption of the host microbiome, or inadvertent administration of endotoxin or other impurities in the bacteriophage preparation. And we're fairly evenly split with 1, 3, and 4 on this one. And again, we'll get back to this as we move forward in the morning. The final pretest question is this one. Where in clinical medicine might bacteriophage therapy find a niche? Which one of these is unlikely, at least over the near term? Okay, again, number 4 is the number 1 choice, and we'll get back to that as we move forward. Okay, so why do we care about antimicrobial drug resistance? Those of you who are in the hospital and in practice realize that over the last couple of decades, it's become increasingly difficult to treat a number of bacterial pathogens. The impact across the globe from the US to resource-limited settings is tremendous, with societal costs in the billions of dollars and thousands and thousands of deaths every year. When you look at what's happened in terms of antibiotic development over the last 80 years, we started out on a rapid pace in the 30s, 40s, and 50s. As we'll discuss in a minute, in the 1930s, antibiotics and phage therapeutics were neck and neck in terms of which one was going to be emphasized for bacterial therapy in man. For a long time, new antibiotics developed kind of every several years, and everybody's optimistic that we'd be able to eventually conquer all bacterial pathogens and put a big red check on infectious diseases and move on. Those who were following, though, what was going on in the field realized that as antibiotics were developed, not too long thereafter, we began to see patients with bacterial infections that had previously been sensitive to the antibiotic placed in the field that were now resistant. In other words, Darwin was right. You can see that by the early 1970s, new antibiotic discovery began to slow and resistance continued to develop, so that by the beginning of the current century, we were seeing more and more drug-resistant bacterial infections and less innovation in terms of the traditional antibiotic pipeline. So how did I get involved in bacteriophage therapy? It was really kind of a serendipitous interaction that involved a colleague of mine from the UCSD faculty, Tom Patterson, shown here, in this November of 2015, when he went to, with his wife, Stephanie Strathty, to visit Egypt on a vacation over the Thanksgiving holiday. They spent some time down in down around Cairo and Alexandria, and after several days there, they got on a barge and floated up the Nile toward Luxor. And on the way, he developed abdominal pain and began to have some vomiting. And Stephanie contacted me by text and said, "I'm concerned about Tom. I think he got some food poisoning here on the boat, and I'm just not sure what to do." As I talked to him more about this, what really was predominating was abdominal pain, not vomiting or diarrhea. Tom was diabetic. He drinks a bit, and he'd had intermittent gallstones. I began to be concerned more about pancreatitis or biliary tree disease than I was about food poisoning. Stephanie had remained healthy. And I told him that I thought it was important for her to get him to a doctor, and she did. And by the time she got him to Luxor, he was close to being in septic shock. When they did a evaluation of him in Luxor, they found that he indeed had pancreatitis. He developed a large pancreatic pseudocyst, shown here, which was posterior to his gastric pouch. After being stabilized in Luxor, he was air-evac to Frankfurt, where he underwent gastroscopy. The pseudocyst was punctured through the gastric wall. And what was isolated in the initial puncture was a multi-drug resistant Acinetobacter infection that had colonized the pseudocyst, presumably while he was traveling up the Nile. The organism was sensitive really only to minocycline. He was given some antibiotics and fluids in Frankfurt after about a week, flown back to the US, where he had a 4-month course over a series of ups and downs with multiple courses of antibiotics in our hospital, essentially getting progressively more and more ill over time. He ultimately ended up obtunded in the ICU with incipient renal failure, intubated, and with discussions going on about limitation of care with his wife, Stephanie Strathney. Stephanie is a very determined person, and she continued to try to think of out-of-the-box approaches to treat him, and just happened to see a new publication that had appeared in PubMed in her Acinetobacter tag about bacteriophages that had been isolated in Georgia, not our Georgia, but their Georgia, that had activity against Acinetobacter baumannii. Stephanie got in touch with me and said, they used to use these, I heard, for bacterial infection. Do you think we could use these in Tom? And at this point in time, we clearly had made very little progress in 4 months. I said to Stephanie, I can't really give you a strong reason why you should stick with what we've been doing, and very happy to see if we can come up with some phages to be able to treat Tom. At the time, thinking it was going to be a real stretch to be able to pull this off in the time that we had left, because as ill as he was, I would not have been surprised on any given night to get called and told he'd arrested and couldn't be resuscitated. Let's go back and talk a little bit about what bacteriophages are. This is a fellow named Harrell who had a very distinguished mustache that was one of the 2 groups that discovered bacteriophages. These initially were described as factors. It wasn't clear they were viruses, but factors found in river water in the Indian subcontinent that could be isolated by putting the water through filters and then taking the filtrate. The filtrate was, the filters were small enough to allow things smaller than bacteria through, but not bacteria. So you could sterilize the water by putting it through a filter. Then if you plated this water on top of bacteria that had grown from that river, holes would appear in the agar on which these bacteria were growing. Something was eating the bacteria. Looked very much like these agar plates. You can see here is a plate by Frederick Tort, the other fellow who was working on this, in which there's a bacterial lawn that had been plated on the plate, and then drops of water had been dropped onto the plate where holes appeared. Bacteriophage means literally bacterial eater. It took a while to sort out what these were all about, but over the course of the next 10 years or so, it became clear that what these were viruses. There are a wide variety of bacteriophages in nature. About 10 to the 31st bacteriophages on the planet, more than any other form of life, depending on how you classify viruses. Their general approach to their prey is quite simple. They attach to their bacterial prey by specific receptors, and most phages have a very narrow range of bacteria they're able to lyse. Once they attach, they then are pulled close to the bacterial surface, fuse with the bacterial surface, and inject their DNA. The DNA, as I'll show you in a few minutes, then is transcribed into RNA. 100 or thousands of phages are made in any given infection over the course of 20 or 30 minutes. Lysins are phage products that are produced by the phage that blow up the cell wall of the bacterium and allow the phage to go on and attack the bacterium next door. These phages were sold commercially for a number of years for treating bacterial infections. You can see here an ad from France about the use of bacteriophages that can be bought for various purposes. Now, the difference between these phages and the ones we'll discuss now is they didn't really understand the bacteriology very well. And you can see from these these titles that they were selling phages based on the syndrome that you were treating. For example, diarrhea, colophage or intestiphage, or for furuncles, staphophage, or just phlegmons, pyophage, but without much effort to really understand the underlying microbiology. It was unlikely that most of these phages, which were just mixes of randomly created phages grown on mixes of bacteria, it was only really a Russian roulette as to whether or not the phage in any given prep were even alive by the time they were sold, or if they were alive, whether they were active against the particular bacterial infection. So over the course of the first 30 or 40 years of phage therapy, both in the US and more in Europe and in the Eastern world, therapy was empiric. The phage preps were not very well characterized. The phage products, which, as I'll show you, have to be made in a specific way, were just kind of filtered out of rough supernatants from bacterial liquid cultures, and with who knows what in the supernates in addition to the phage. As I mentioned, they were used syndromically, and there was very little evidence of any microbiological effort to assess what they were doing. Patients were classified in research projects as to whether they got better or not on a subjective basis, not whether or not a given infection was cured by looking at microbiological parameters. So over the course of about 30 years, they were gradually discredited in the West, and starting from about the 1940s on, we went in the direction of antibiotics. But in the East, and by that I mean Russia and the former Soviet republics, bacteriophage therapy is being used to this day in very much the same way as it was used in the earlier days, very empiric and just beginning now to be used in a more directed way. Against that backdrop, we sought to find phages that would be active against Tom's organism. Through a kind of a long, shaggy dog story, the phage that Stephanie had seen reported in the journal article I showed you had found its way through Belgium to Ry Young's lab at Texas A&M. Ry has a very interesting phage biologist who, for his entire career, has been working on phage biology and thought it might be interesting someday to treat patients, but had always been reticent to actually give them to humans. Stephanie got him on the phone one Friday night, and after about an hour and a half of talking about how her husband was dying and something really needed to be done, Ry said, "Okay, fine. I've got a couple of postdocs who aren't doing this weekend. Send me the organism. I'll see what I can do." And so he began to go on a phage hunt, trying to find phages that were active against Tom's organism. I, in the meantime, had been in touch with the FDA because I knew we'd have to obtain an IND to give the phages. And the FDA reviewer I got, rather than being obstructionist, said, "You know, this is really interesting. Most of the time what I do is stool transplants, which is really pretty terrible. This is actually quite good science. And I know about a group over at Walter Reed at the Naval Medical Center that are working on phages for Acinetobacter. Here are their phone numbers." We got in touch with them. The ones at the Navy in particular had a whole library of phages that were active against Acinetobacter, and they too agreed to screen Tom's organism for phage that were active against his A. Bellmani. And over the course of the next couple of weeks, both labs found 4 active phages. And in a process that was a bit of a scramble, we were able to purify them to the point that we were able to give them to Tom as his infection progressed. The Navy phages arrived, the Texas A&M phages arrived first, and because they really hadn't been given to humans before, under an IND from the FDA, we decided to give them into the abscess cavities in Tom's stomach. At this time, he had 5 abscess cavities that were being drained through catheters that were being irrigated every few hours. This is Andrew Peisel, the interventional radiologist who is irrigating Tom's cavities with phage. Over the course of the next couple of days, not much happened. He didn't get any worse, but he didn't get better. He'd had periods of stability for a couple of days. But he wasn't getting better. And when the Navy phages arrived, we decided that since the organism was being found obviously in his abscess cavities, but also freely in his peritoneum and his sputum and his urine, intermittently in his blood, that he had a systemic infection. We weren't sure where the phages were going. We decided to give the phages from the Navy by an intravenous infusion. This is our ID fellow giving the phages intravenously about 2 days after the Texas A&M phages began. Over the next couple of days, Tom actually began to stabilize. His pressor requirements declined, and he became much less obtunded. By Saturday night, was awake and recognized his daughter. And after a very rocky course that lasted several more months in the ICU, he gradually improved with less and less Acinetobacter growing from various orifices and recovered to the point that he has now returned to being on the faculty at UCSD. This is Tom with Theron Hamilton, the Navy scientist who came up with the phage cocktail for Tom. Now, for those of you who are actively reporting me as a HIPAA violator, the real HIPAA violator is Tom and her husband. They've written a book, or Stephanie, her husband, they've written a book about this that goes in a bit more detail about some of the scramble to get this all done. But what this really did was gave us some insight into what the challenges were and what the opportunities were with phage therapy that I'll go into for the next few minutes. So what did we learn from this experience that was different from what we learned in the past? First was that it was very important to be able to have phages that you knew were active against the patient's organism. This is an approach that the Navy uses to choose phages that are active against a given organism. They place the organism in a 96 well plate, in each well of a 96 well plate, and then in each of those wells using robotics, they put different phages in each well, and they look to see which wells, in which wells the metabolism of the bacterium is retarded. And you can see that in this experiment, there are 5 phages that suppress growth of the organism over the course of a 20-hour period. And then when the phages are put together in a cocktail in the mixture, same thing is true. This is a depiction of the 98 well plate, and 96 well plate. And what you can see is that in dark boxes, there were 6 phages that were able to suppress growth of the organism for over 20 hours. These phages were at the time thought to be different because they were screened against a couple hundred different Acinetobacters, and they had different levels of activity against different subsets of Acinetobacter. Acinetobacter happens to be an organism in which any given phage is likely to infect only a small sliver of the Acinetobacter. And so at the Navy, they thought that because any phage in their library happened to attack different subsets of Acinetobacter in their library, they must be different phages. They hadn't yet sequenced them. But we chose 4 of these and put them together in the Navy cocktail that I mentioned. Now, how were they purified? They're purified and prepared relatively simply. If you kind of take a flask of bacteria, shown here schematically, and you add the phage that you've selected as being active against the patient's organism, they replicate in the bacteria and knock them off. And then over time, what you end up with is a bunch of dead bacteria and many more phages than you started with. And the problem you have now is separating the phages from the dead bacteria, endotoxin, and other things that, that go with the preparation in this big reactor. The way you do that is to create a chamber that has a filter that's able to filter out the bacterial, living bacteria and bacterial schmutz, if you will, and then pass the material through this, and you end up with purified phages down below, but also endotoxin and a bunch of other molecules that could cause toxicity in patients. At this point, you then have to purify the endotoxin or the phage preparation by removing the endotoxin and other products by ultracentrifugation. You can band centrifuge the phages. You can dialyze away some of the contaminants, or you can use organic solvents to extract them. But through different methods, depending on the phage prep you're using and what impurities you are, you can end up with phages that are actually extremely pure that allow you to give large numbers of phages with very little toxicity. The only toxicity that we generally anticipate from phages are if this process fails and you end up with endotoxin, one can have toxicity from that. But because we have very good assays for endotoxin now, this is really not an issue when these are done with current methodologies. Now, the next thing we learned is that as with bacterial pathogens, resistance to phages also develops. This is the graph I've already shown you, showing you the way that the phages used for Tom's organism suppress growth for 20 hours in this 96-well plate. This is the same experiment, but with the Texas A&M phages. You can see they're a bit leakier. They're beginning to allow the bacterium to grow about 10 or 12 hours out. Now, 6 days later, you can see that an organism obtained from Tom was now fully resistant to the Texas A&M phages and was beginning to be resistant to the phages from the Navy. And by 10 days, resistance was complete. Now, over this period of time, he had clinically improved substantially, although the phages were no longer active. Now, as I mentioned, we thought that we were going to try to get around this issue of rapid development of phage resistance because we thought that we had different phages and the organism would have to work around multiple different mechanisms of action. It turned out later when these phages were all sequenced that they were very similar to each other. This is a dendrogram of kind of the phage world of Acinetobacter phages. And you can see that the phages from the Navy were all right here on the same tip. They just happened to have very subtle differences, we learned later, in how they bound to the same receptor, so that they were rapidly selected against when they were used to treat Tom. The Texas A&M phages turned out to be very similar. Now, again, one of the nice things about phages is you can go back to organisms that develop resistance, like the ones down here obtained from day 10, and rescreen them with the same library, looking to see if you can find phages that will suppress the drug-resistant bacterium. And when we did that, we found that there were new phages that were able to now suppress the resistant bacterial mutant, and we could go back in and treat the patient with these new phages, which is what we indeed did. And these are kind of the mug shots, and this new phage that had activity is shown over here, very different genetically, we found out later, from the ones we've been using. So the principles of therapy are very much like those with antibiotics, in which resistance can be selected. Look for orthogonal resistance patterns and try to treat the patient with agents that have orthogonal resistance patterns. We've been doing this in antiretroviral therapy and HCV therapy as well for the last several decades. The phages that were used here were 4 myophages. They're called myophages because they have this long tendril, looks like a muscle that then pulls the phage into the organism when it attaches. These potophages, the little short feet, this is the one that was selected for after resistance developed to the originally treated bacterium. Now, one of the things we also learned was that when bacteria develop resistance to phages, they often have to give up something to be resistant to the phages that you're using to treat. Very much like the fitness cost we see in antiviral therapy when viruses are, for example, selected to be resistant to 3TC, they're a little bit less fit than wild type when growing outside the selective pressure of 3TC. In the case of these phages, what we found is TP1 was the organism pretreatment. You can see these big juicy capsules around, as a halo around the organism. This broke down and degraded over the course of as you went through the isolates. And so what these phages were doing were disrupting the genes that specified, what the organism was doing was being selected for bacteria that no longer express the cell wall to which the phages could bind. And so we now had less fit bacteria because the cell wall was gone, but they could still grow, that were resistant to the phages that were being used the first time around. These bacteria, when put into an assay in silkworms that is often used to look at bacterial invasiveness, were much less invasive than these. So the trade-off was actually to the host's benefit. And what we're, I think, increasingly learning is that many of the principles of therapy that we've used for traditional antibiotics and for antiviral therapy are ones that are going to come to play in phage therapeutics. Now, let me shift gears and talk about another area of phage therapeutics that is evolving relatively rapidly, and that is specifically genetic engineering. Most of the phages that have been used therapeutically in the past are ones that have just been found from the environment. People go out and look for phages in sewers and places that bacteria grow to look for phages in the environment. Sometimes you can find phages that are perfect. Sometimes the phages aren't quite perfect and may have features that make them less suitable for use therapeutically. For example, carrying bacterial resistance genes or carrying genes that allow the particular phage to integrate into the DNA of the bacterial cell, which is something that you don't want it to do. Graham Hatfill, who is a colleague of mine from the University of Pittsburgh, is an expert in phage biology for mycobacterial phages. He's been running a program in US undergraduate schools with support from the Hughes Foundation and the NSF, in which undergraduates isolate phages that are active against Mycobacterium schmegmatis, a non-pathogenic mycobacterium found all around us. They characterize the phages, they sequence them, they name them, and then they mail them to Graham. And Graham has 15,000 of these he's collected over the years, frozen in his freezer. He found out that some of them had activity against other mycobacterial, including Mycobacterium abscessus. Some are even active against M. tuberculosis. And I began to talk to him about whether or not we might be able to use these to treat patients who had mycobacterial infections, for example, M. abscessus, in transplant patients or patients who had cystic fibrosis. One of the patients that came along was a young woman from the UK who had severe cystic fibrosis, who'd been carrying an M. abscessus infection for about not quite a decade that led to progressive pulmonary dysfunction. She had to undergo a transplant, lung transplant, did well initially, but then 2.5 months post-transplant, the infection came back. She was treated with antimicrobial drugs against her organisms. Efforts were made to reduce her immunosuppression. To make a long story short, she developed lesions along her sternal wound and showers of skin lesions that were biopsied and found to be granulomatous infections. In other words, disseminated mycobacterial disease. She was sent home on hospice. Here are pictures of her cutaneous lesions that were biopsied. This is what was going along on her sternum. And here is kind of hot activity in a number of lymph nodes. So Graham, as I mentioned, was running this program called Phages that provided him with this freezer full of mycobacterially active phages. But the problem with these phages is that, like all mycobacterial phages, they have another quality that some phages have called lysogeny. I've been talking so far about phages that have this very simple lifestyle. They infect an organism, they replicate, they make lysins, they blow up the organism, they do it again. Under conditions of nutritional deficiency, in other words, when the organisms are stressed, instead of phages going in and knocking off the organism, some phages have two enzymes, one an integrase and the other is a repressor gene that allow the phage to integrate its DNA into the DNA of the host bacterium. And then as the bacterium replicates, the phage replicates with the bacterium. And then periodically, the phage will be induced or be reactivated. And then go into a lytic cycle, and go on to infect other organisms. The problem with giving phages that have this characteristic of being able to be lysogenic is that when they integrate into the host DNA, they can take with them as they come out genes that specify bacterial pathogenicity, genes that specify drug resistance, and the repressor genes they use to get in and to stay in these organisms also keep new phages from being able to attack them. So what you would like is to have a phage that can infect your organism, but doesn't know how to go in this direction. And so what Graham has done is developed an approach to be able to genetically engineer phages and take out the genes that allow the phages to become lysogenic. And what he essentially does is he takes phages, puts them into a permissive mycobacterial organism with and electroporates in segments of DNA that he wants to excise from his organism. And what happens is that some of the colonies of these mycobacteria in which both the phages and this deletion catalyst have been placed will delete the area you want out and no longer are able to be lysogenic. And so what Graham has been doing is developing approaches that allow him to instruct the phages not to go in the direction of lysogeny, but to only be able to have this lytic lifestyle. So he was able to take this approach and had several phages that were actually active against the isolate of our patient from the UK, began to use them. And again, she began to improve over the course of about a week. You can see here her cutaneous lesions melted and went away. You can see that her hotspots, which we think were granulomatous infections of lymphoid tissues, also began to go away. And you can see that her pulmonary function test, which had kind of plateaued post-transplant with phage therapy, continued to improve. She did very well for a long period of time. She's recently developed a relapse. We're trying to find new phages to treat her with, but she was able to return to school and did well for 2 years. So what we found here is that you can actually genetically engineer phage. We're allowed now by regulators to be able to give genetically engineered phages to humans, which may allow us to tailor receptors and do other things that will allow phages to be more broadly used. So let me just, as we're beginning to wrap this up, talk about other potential clinical niches. I've been talking primarily about using phages to treat mycobacterial infections and bacterial infections that are resistant to traditional antibiotics. But there are other kinds of approaches in which phages may have clear clinical benefit. This is a paper put together by my colleague from UC San Diego, Saima Aslam, who's taken this phage therapeutics area and run with it and has treated quite a few people now, both here at UCSD and through consultation elsewhere with different clinical indications. I'll just highlight a few of these in our first 10 to give you a sense of some of the things we've learned from these patients. This is the patient I've talked to you about already today, Tom Patterson. The first patient she treated was a patient who had had a lung transplant for idiopathic pulmonary fibrosis. There was a complication of his anastomotic site of his trachea, and he didn't clear secretions well, had a series of drug-resistant Pseudomonas infections that got presumably increasingly worse to treat. And what we learned from him is that phages find out where the infection is and go there. And what do I mean by that? We found some phages that were active against his organism in the hands of one of the biotech companies that were working to develop phages therapeutically. They wanted to give the phages by an aerosol route. We wanted to give them intravenously because we weren't certain about how the phages would be delivered if we tried to give them by an aerosol route. When they arrived in our hands, we spent some time trying to understand which nebulizers would not destroy the phages. That took several weeks because many of them actually have enough shear that they will destroy phages. And in the meantime, they began to treat the patient intravenously. What we found is that about 48 hours after the first phage infusion, when he was bronchoscoped, intravenous infusion, when he was bronchoscoped, his trachea was loaded with phages at much higher levels than one could calculate had been given to him when he was treated, with a number of phages that were given intravenously. So one of the nice things about phages from the pharmacologic perspective is they're kind of the inverse of antibiotics. We always worry with antibiotics when we give them about how fast they decay in the lung or the wound when we're trying to treat pneumonia or wound infection. What phages do is they find where the infection site is and they replicate there. So they're really kind of self-replicating antibiotics. Another patient who was treated was a man who had a left ventricular assist device that had become infected after transplantation. He had Simon has now had several of these. This one was Pseudomonas. She's had another patient who had a staph infection. And in these two cases, what was exploited was the fact that we were unable to clear the infection from these left ventricular assist devices with traditional antibiotics because they were protected by, the bacteria were protected by biofilms. And phages actually are very disruptive to biofilms. And one of the things that phages are good candidates for is to be used along with antibiotics to disrupt the biofilm and to allow the successful treatment of infections that are otherwise couldn't be reached because of the biofilm that was created on an implanted device. So there have been a number of infections now treated by Simon and now others that are involved in implanted devices such as left ventricular assist devices, joints that with phage therapy have been successfully treated. Now, what we've done over the last several years, and by we, I'm talking about Stephanie Strathy and myself. Stephanie, as you may remember, is the wife of Tom Patterson and a member of the faculty here as well, infectious disease epidemiologist, put together an institute called IPATH, which is a phage therapeutics institute at UC San Diego. And we've been providing assistance to people who are looking for phage for clinical use. And over the course of the last number of years, we've been trying to learn about how we can best take what we've learned and treat people with bacterial infections. What we've learned is we have a long way to go to be able to treat people in a reliable way because of a number of bottlenecks in creating phages that can be used for clinical use. And the bottlenecks are really outlined for you here. The first is you have to find phages that are active against a patient's organism. In some organisms, it's quite easy. Phages that are active in the area of Staph aureus, for example, are often active against most staph you come across. So you can have a handful of 10 staph phages in your library, and almost any staph isolate you run across, you've got phages you can go after the patient's isolate with. On the other hand, with Acinetobacter, any given phage might only treat 2% of all the Acinetobacter you run into in clinical use. You'd have to have a couple hundred phages in the library to be able to find phages that are useful there. Once you do that, you then have to sequence the phages and look for evidence about whether the phages have any ability to be lysogenic. Can they integrate into the DNA of the bacterial host? Do the phages themselves carry any toxins or virulence factors, antimicrobial resistance genes, and look for contaminants in the process that you're making? And these kinds of phage library constructions can really make it difficult to find a given patient a phage unless you have a library at hand, which is what we're gradually trying to accomplish in our center and other centers around the country. The experience we've had is that the organisms that we've received requests for assistance turn out to be ones you might expect, organisms that are often drug resistant, Pseudomonas and E. coli, carbapenemase producing Enterobacteriaceae, with some mycobacterial infections. And the success in being able to find lytic phages really depends very much on the organism itself. We do much better with, once we start looking for a phage with organisms like Pseudomonas, which are kind of closer to staph than Acinetobacter. And we do pretty well with organisms in which the phage library is relatively broad. So where do we go from here? Well, we obviously need broader libraries of phages. We were very lucky with the patient that I mentioned to you who had the M. abscessus infection. Graham now has thousands of phages, but only has been able to find phages that are active against about a third of the isolates that we confront in clinical medicine. He's trying to figure out how to broaden his library. We don't yet have phages that are reliably active against Burkholderia. These two organisms themselves are really major problems for people with cystic fibrosis. We need to go from anecdotes, which is what this was, to doing rigorous clinical trials, very much like antibiotic development. So we'll be able to understand how phages can best be used in clinical medicine. One thing I didn't talk about is that when you give phages either topically or intravenously, you can develop antibodies to the phages. These are not toxic to the host, but they cause phages to be cleared from the bloodstream more rapidly. And one of the issues that we have to think about is how these do or don't affect phage efficacy. Some animal models suggest that this is a killer for a phage efficacy. Other ones don't. It may well depend on the size of the bacterial population you're trying to treat, how well protected it is from the antibody compartment, and whether or not what you've done with the phages occurs in the first week or so before antibodies develop. It's more of a problem in immunocompetent patients than it has been in, for example, transplant patients where this has really not been an issue. But one of the limiting factors that we have to think about with phage therapies. It'll be a while before we get to being able to do this at clinical scale, but I do think that over time, one of two different approaches may well be developed. One would be fixed phage cocktails that essentially involve taking a known group of 3 to 5 phages that are active against most organisms of a given type. For example, Staph aureus, which, as I mentioned, has a relatively limited phage repertoire. In this case, if you have the right 5 phages, kind of like the right antibiotic, when staph comes along, you can use it to treat a patient empirically, and then test in the laboratory whether or not the antimicrobial, the living antimicrobial have, is active against the organism you're treating. The downside of this approach is that not every phage in your cocktail will be active, and you may find yourself selecting for drug resistance. The upside is you can already have this in your hands, have it in the hospital formulary, because these are quite stable once made and stored, and use them just like you would order up an antibiotic. The other approach are the custom phage cocktails. For this, we have to rely on these for organisms like Acinetobacter, in which any given phage is unlikely to be able to lyse any given bacterium. And you have to have a large collection of pre-characterized phages that have gone through that selection process I mentioned before, sequencing and characterization, already kind of on the shelf and ready to be screened for the organism of the patient you're treating. You can screen them robotically like they did at the Navy, and then pick the active ones and use to treat the patient. And the benefit here, of course, is that the phages that you make in the cocktail are going to be active against the patient organism that you're treating. The downside is this takes a bit more time. It could be done as shorter period of time as 48 hours or so if you already have the phages in your hands, but it will take longer if you have to go back and find these in nature. So where are we now? Over time, where we're moving beyond environmental phages and beginning to engineer phages that have organisms that have characteristics that allow us to be able to engineer their receptors so they're able to attack a broader array of bacteria of a given species. So we might be able to have more fixed phage cocktails. Phages carry a warhead that allow them to knock off the lysine, the lysine warhead. You can engineer the lysins to be more toxic to bacteria. People have been able to engineer phages to include CRISPR mechanisms to knock off bacteria. And as I've mentioned already, Graham Hatfield has found ways to make lysogenic phages only lytic, so we can engineer the lifestyle of phages with synthetic phages. And we're at the point now that companies are doing this, and we're headed in the direction of being able to use this clinically. So in terms of where we're going with synthetic phages and with these engineered phages and clinical investigations, can be very much like antiviral therapy was. We spent a lot of time with rigorous clinical trials that were, that in parallel, we looked at things like how lytic the antiviral drugs were. In other words, what did they do to the viral load? We're doing the same sorts of thing with phages, looking at how they lyse organisms in situ and in the laboratory, going through these clinical trials, refining the phages, coming up with better approaches, developing iteratively optimized phages, and learning better how to use them so that over time, we may well move to synthetically derived phages with the principles of therapy worked out very much like we have with antibiotics, and the ability to use phages in clinical medicine in parallel with antibiotics and in conjunction with antibiotics to provide us ways to go after more bacterial infections that we're having difficulty treating. So I'll stop here and we'll go back to our pre-test questions. As I mentioned, we have this Phage Institute at UC San Diego. You can find us on our website, ipath.ucsd. If you have any questions about phages or patients you'd like to refer, we're glad to help you as best we can. So let's go ahead and go through these post-test questions. Jose has put up the questions for you, and why don't you go ahead and see where you stand after these. How do phages kill their prey? And we'll see what the answers are after my droning and overly long lecture. How's the polling coming, Jose? Do you have enough to show us? Okay, so the real answer here is this one. They were able to do it best by being able to create license. They can integrate their DNA, but when they do this, they actually help the bacteria live because they create, they don't really downregulate any of the bacterial host RNA, but they do prevent the bacterium from being invaded by other phages. So the correct answer here is they create license that disrupt the bacterium. Let's go on now to the next question. What are some of the toxic side effects you might expect with phage therapy? Can they induce septic shock by killing every bacterium within the organism, within the patient right away? What about the induction of antibody responses? Can they massively disrupt the microbiome? Or could you inadvertently give endotoxin that could be toxic to the patient? Okay, so the best answer really is the last one. In practice with our new approaches, this is really not a big issue. The other issue that's important to know is when patients have disseminated bacterial infection like the one we had already, they're already awash in endotoxin, and they are tachyphylaxed endotoxin. So if you did have a little endotoxin in the prep, it would be unlikely to have an impact on the patient. Patients who aren't seeing endotoxin, however, can get into real trouble if you don't go into the purification process that I mentioned. Antibodies do develop, but there's really no evidence of any toxicity to the patient. The antibodies may have an impact on phage longevity in the patient, but we haven't seen issues, for example, with acute reactions or with serum sickness from this phage response. We're awash in phages all the time ourselves. We develop antibodies to them, they're cleared, and we don't have these toxicities in non-therapeutic settings. This is a great thing to talk about because, in fact, they kill bacteria extremely well, but so do antibiotics. And in some very nice experiments done by a phage biology group in France, in which they took antibiotics or phages and titrated them so they killed their bacterial host just as rapidly by looking at kill curves. Less endotoxin was turned out by the phages than by most antibiotic interactions. So there's no nothing that antibiotics do that phages are doing. And this is exactly right. One of the beauties of phage therapy is that you're only killing a sliver of the bacteria that a patient is carrying. Unlike antibiotics, which are kind of like throwing a cherry bomb into the microbiome and other things fill in what you've left the hole for, phage therapy is really much more like a laser. And in theory, will give you less problem with ongoing selection for drug-resistant organisms. And the last pretest question is here. What are potential clinical niches for phage therapy? Implanted devices, multi-drug resistant pathogens, mycobacterial infections, fungal therapy. Okay, absolutely right. There's still quite a bit of work to be done on antifungal therapy. We don't really have phages right now that reliably that have been worked out in this realm of biology much further down the road. So let's close that and we can go on to the question and answer period. I appreciate very much your attention, and let me begin to, you can use your Q&A button. I'll go and look at some of the questions that are coming along here. So the first question by Dr. Khan is, can antibiotic resistance be tackled by phages? In fact, they can be. There some very nice work done by Paul Turner and his colleagues at Yale, in which phages were specifically chosen to attack the pumps that pump antibiotics out of their bacterial hosts. And by blocking the ability of antibiotics, of bacteria to pump antibiotics out of the bacterial pathogen, they were able to resensitize the bacterium to the antibiotics that were being used to treat the patient. It was used very successfully in a man who had a very large dacron aortic graft that was infected with Pseudomonas that couldn't be removed. But by using a phage that shut out the pump that was effluxing the antibiotics, the synergy allowed the patient to be cured. Dr. Khan also asked whether phages can infect humans. The reason they don't infect humans is because the receptors that they are bred to, that they are selected to in nature, are ones that are found on bacterial cells. And we have enough of viruses on our own that we really don't have phages that attack human cells. What happens when the bacterial pathogen clears? Dr. Douglas asked that great question. In fact, that's exactly what happens. When there's no food, there's no phage, and the phages are cleared by the RE system. Normally, if you give phages intravenously for which there are no bacteria in the food chain in a given patient, they're cleared out by the RE system and chewed up in the liver, spleen, and excreted. And when food is no longer present, off they go. So they're naturally cleared. David Butler is asking about selective pressure, evolution of the bacterium. Can you actually, do the phages chase the bacterium? You know, clinically, that's a great question, and one of the things we weren't able to investigate in Tom, but in nature, that's exactly what happens. And I think as we begin to use this more broadly, we should look more carefully at what this happens. In vitro, you can show that that happens, that if you dab phages onto a bacteriological lawn, they will chase the bacteria as the bacteria develop resistance and evolve that way. Dr. Ferris asked about international collaboration, about whether there are efforts to create international phage libraries. There are a lot of discussions going on now with Eastern Europe and Georgia. They have immense libraries. What they don't have right now is the ability to to purify them to the extent that they can be used the way that we're using them in the West. And these sorts of collaborations are ones that would be really quite valuable. They do sell these phages in the West. They're mainly sold to be used almost as nutraceuticals because they generally are not well enough prepared to be given intravenously, and an alliance like that would be really quite, I think, in the long term, beneficial. We don't have, in answer to Dr. Kressel's questions about phages for fungi, right now we don't have them for fungi. Dr. Gill asked about for mucor. It'd be great if we did, but the answer is no. Dr. Kahn is pointing out that we're all vexed by the same autocorrection and autocorrectors like phases rather than phages. So we'll have to train our autocorrectors to learn what phages are as well. Dr. Ording-Bauer asked about other mycobacteria. The question's a great one. And right now, phage selection is empiric for the most part. We don't completely understand how to select phages based on molecular analysis of their putative receptors. There's a lot of work going into informatics to try to learn how to do this. In Graham's particular collection of phages selected in the wild for M. spagmatis, he has some that are active against Mycobacterium abscessus. He has almost none that are active against Avium intracellulare. Some are active against M. tuberculosis. And he can take some that are partially active and by serial passage, train them through host adaptation to be more active. But why it is that the phages selected in M. spagmatis are not active against some species of organism, I think really is not yet clear. Do these other organisms have their own phage lives? Of course they do. And I think that if one went to the same extent to try to find phages against them in nature, libraries could be developed as well. Do phages establish residence in the microbiome of the host? Dr. Sag asked. If so, they persist at the site of infection and how long? Well, they'll persist in a host as long as there's something to eat. And if you look in the GI tract right now, our GI tracts are loaded with phages that are in a dynamic relationship with the bacteria in our GI tract that turn over almost continuously. So we are kind of living in an equilibrium in our GI tract with the phages that are resident bacteria there. People have even raised questions about there are phages that will attack C. difficile. One of the problems with them is that most of them are lysogenic. Martha Clokey is working on developing phages that are just lytic against C. difficile. But people have actually looked at fecal transplants and looked at fecal transplant filtrates and sometimes have seen benefits from that, making one wonder whether or not it's the phages that are doing some of the work as opposed to the bacteria in these transplants that allow reconstitution of a normal, more resilient bacterial flora. Dr. Keller asked about if there is a patient with pan resistance at an outside hospital, is there a way to apply for access to phage? There absolutely is. Please get in touch with us through the website. The way the phage world has worked is that there are a number of different groups around the US and around the world, each of which work with kind of a subset of phages. There's a group in Texas, Dr. Anthony Moresso's group that does a lot of work with E. coli and Klebsiella. There's a group at Yale, led by Ben Turner, or by Ben Chan and Paul Turner, that have a big Pseudomonas library. We have phages here that are active against staph and Enterococcus that Dr. Pride is working on. So what we will do is kind of serve as a dating service to try to help you find a laboratory that's willing to look for a phage for your patient. So we're more than happy to help with that. Dr. Canoni has asked about bacteriophages and CNS infections. We do know they get across the blood-brain barrier. We've only had one patient that we tried to treat with a CNS infection. It was a patient who had infected a large plate used to reconstruct his skull after a traumatic injury. He had developed meningitis with Acinetobacter. We were able to treat him for a couple of days before his family decided to go for comfort care. We were able to show that the phages that were given intravenously got into his CSF, but unfortunately, he wasn't treated long enough to know whether or not we were able to, whether it had an impact on him clinically. Dr. Ferris thanks Stephanie and the IPATH team for help. We're more than happy to help our Canadian neighbors and look forward, and we're really very much appreciate your willingness to allow us to visit you again. We hope that inverse is also true soon as we try to get that border open. And then let's see, which are the bacterial diseases where bacteriophages have been used? Basically, many different types of bacterial infections, ranging from staph to gram-negative Enterobacteriaceae to Pseudomonas to mycobacterial infections, all of which have phage lives. There are some organisms for which phages have been more difficult, mainly because of libraries that are available. Burkholderia is a good example of one that we wish we had more phages for, and I've already mentioned some of the mycobacterial problems. Dr. Spear asked if we can predict if a patient will generate antibodies to phage therapeutics and how they impact therapy. If a patient is immunocompetent, they have a pretty good chance of developing antibodies over a couple of weeks. Virologists and immunologists have used phages for a long time to study the evolution of neutralizing antibodies in humans in immunology research. And we know that you can measure antibodies in the bloodstream over a period of time, and that when you measure phages in the bloodstream in people with antibodies to the phages persist for a shorter period of time in the bloodstream. Without antibodies that you can measure, if you inject phages, the half-life in the plasma is 30 to 60 minutes. When you do when the patient develops antibodies, they're cleared more rapidly, but they still can circulate long enough to get some sites of infection. In some animal models, you can show that you can treat an animal with a phage, for example, for a wound infection successfully. You can take that same animal model and immunize the animal with the phages you're going to use, and then treat the animal in the presence of those antibodies from the get-go and still have success. That doesn't happen in all models, though, and Dr. Hatfull has some interesting experiences that he's published in non-transplant patients with Mycobacterium avium infections who developed antibodies that interfered with phage efficacy. In other words, initial success and then antibodies developed. So it's an area that we need more work in. It happens more often, but in people who are immunocompetent, we often have these infections in people who are not, and we're still trying to figure out how much they limit therapy in real life. And Dr. Park has a question about anecdotes with these questions n equals 10. I'm not sure what your question is. If you're talking about the paper that Dr. Aslam wrote, the paper is in Open Forum Infectious Diseases from last year, and each one of those papers, patients in the table that I showed you, there are more details about the patient in that paper and in other publications that are referenced in the paper. And I think we've gotten through all of the questions. Are there any other questions that anybody has? All right. Well, thanks very much for your attention and for putting up with kind of an offbeat topic for someone who is more of an antiviral chemotherapeuticist than somebody who treats bacterial infections, but still finds viruses interesting in either direction. So good luck with your ongoing pursuits of making the world more sterile for patients. And I look forward to seeing you again in another seminar. Have a good rest of the summer, and let's get COVID behind us.
Thank you, Dr. Schooley. As a reminder to our audience, evaluations and information on how to claim continuing education credits will be emailed by 5 p.m. Pacific time tomorrow, and this will enable us to review all of those that have attended today's live broadcast. Here's a list of upcoming webinars, and to register, you may visit the IAS-USA website. The next webinar that we have lined up is an update from the IAS conference, which will be held on September 7th. A new IAS-USA website feature, Practice Question of the Week, is now available to help our audience stay informed of the state-of-the-art practices for the prevention, treatment, and care of people with or at risk of HIV, COVID-19, and other viral infections. A new question has been posted today, and here are the results from last week. You are also able to access these questions on the resources section of our website. As a reminder, application to attend the annual Ryan White HIV/AIDS program clinical conference is now available on the IAS-USA website. And here are a list of recent on-demand dialogues. And please keep in mind that COVID-19 information changes rapidly, and the webcasts and podcasts have a limited shelf life. Lastly, we'd like to thank our presenter, Dr. Schooley, and to the audience for your participation. This concludes today's webinar. Thank you.